Hi guys, my name's Loren, and I am at Georgia Tech in Atlanta, and I guess you are all over the world, including even India. So I guess you can ask me questions, and I will get them and answer them, but if not, I will just start talking. And I guess I will start by talking about the work we do, which is focused on the origin of life and sort of ancient biochemistry. So here at Georgia Tech, actually, we have a big effort in this area. We have two centers. One of them is a NASA-funded NAI center, which is focused on the origin of life. Actually, it's using extant biology in order to understand the most primitive biology we can, which we think is very, very primitive. So that's our NAI center focused on the ribosome. The other center we have is headed by Nick Hudd, which is an NSF-funded center, which is devoted to the understanding the origins of informational biopolymers. So the idea of these two centers, in the best sort of in our happiest imaginations, is that our NASA center is drilling down, trying to understand the origin of life, and the NSF center, Nick Hudd Center, is pushing up, trying to understand. And we will meet in the center and drive the golden spike and be able to really understand the origin of life from both sides. And actually, we think we're going to be successful. In fact, in some ways, we think we have been successful. It's almost a matter of explaining it and articulating it to people. But we think we have really, really figured out some things. So, and if you guys are interested, I can explain to you some of our ideas. But first, I guess I will start talking about my center, which is the NASA-funded center, and this is a center focused on the origin of life, and it's using the ribosome. And I guess I need to explain to you a little bit about how that works. Is that the ribosome, and I'm not going to justify all of this right now like it's a technical talk. I'm just going to tell you what I think, and if you don't believe me, you can ask me or we can talk about it. But the ribosome is the operating system of life. It is the thing in biology that is universally conserved. It has been, at its core, it has been constant certainly since the last universal common ancestor. We know that. So it basically was, it predates the last universal common ancestor of life, meaning that it is about minimum 3.8 billion years old, something like that. And I can make a firm prediction, and I'm willing to bet beer or scotch or something that if you wait another 3 billion years, the ribosome is not going to change. I mean, it's done. And it is that core operating system of life. Everything else is basically a peripheral that can be swapped out. You know, most of metabolism can be changed around. DNA polymerases have come and gone. But the translation system and the ribosome is that permanent, unchanging part of biology. And so we use the ribosome as a telescope to drill down and say, how deep does this go? And basically, our current models are that the ribosome, in fact, the more I study the ribosome, the more, in my perception, the older it gets. I mean, I used to think it was one of the ancient assemblies in biology. Now I actually, in our best models, the ribosome really is an ancient prebiotic assembly that has persisted. And that it really is sort of older than life itself. And so it's older than, it depends on how you define life, of course, but I mean, for most of our definitions of life, it's older than catalysis, I can say that. It's older than the components of which it's made, meaning there has been an evolutionary process by which there's been a fossilization process where we now have coded proteins assembled in the ribosome, but those are replacements of a primitive polymer that is older than protein. And the same actually goes for RNA.
Two questions on the
Okay, so I have some questions here.
Yeah, mine is on top actually. I have one.
Okay.
I was asking whether ribosome does, because you said ribosome what we have right now is what it is. Doesn't that make it like non-life because you're implying that it doesn't evolve anymore?
Okay, do I need to restate that question?
Yeah.
So Batul has said, she said, if it doesn't evolve, then is it alive? And you know, isn't that sort of breaking some definition of life? And that's I guess because the standard definition of life is something that, you know, evolves by Darwinian mechanism. And I think that basically is a problem with the definition of life. In fact, my definition of life is anything with a ribosome. And that sounds kind of trivial, but basically what the ribosome is a collaboration of really different kinds of polymers. And so the core of life is this collaboration between polypeptide and polynucleotide. And to say that it doesn't evolve is sort of amazing, but it really doesn't. I mean, if you look at how much the core of the ribosome has changed over the last 3.8 billion years, it does not evolve. And so I would just sort of say, if the ribosome doesn't fit your definition of life, then the problem is with your definition, not with the ribosome. Does that do okay?
Could you read the questions?
Okay. So Sanjoy has a question here. Do we understand how the ribosome came to be? Well, we have models for how the ribosome came to be, and I think we do. And these are experimentally testable models. In fact, that's one of the primary focus of my center is what we call molecular resurrection. We have models for what the ancestral ribosome was, and these models, of course, are molecular, and so we can make them. So we have certain predictions on folding and function of ancient components, the core components of the ribosome, and so we make those. So I think we do have a good understanding of how the ribosome came to be. So the question is, do we understand it? You haven't really asked me how did it come to be, but do we understand it? Yes, I think we do have a pretty good understanding. Yes. And now there's another question. While they're flipping off here, I have the same kind of question as Sanjoy. Do we have any ideas about the prototypical functions? Yes, of the ribosome. Okay, so what did the ribosome, what was its original function? I mean, one thing we know about the ribosome, number one, is that the ribosome today makes coded protein, and it takes RNA, translates it into protein. And we are quite certain that the core function of the ribosome is older than protein, and I can maybe explain that to you, but for right now, let me just say that the ribosome is clearly the core of the ribosome. And what I mean is the peptidyltransferase center is older than coded protein. And so you can say, what did it do? Well, it did a very primitive kind of synthesis. In fact, we call it basically a molecular sausage maker. And our idea for the ancestral ribosome was that non-specific condensation. So it could make short oligomers of peptide, ester, thiol ester, all kinds of just condensations. And the core of the ribosome is not like a normal enzyme. It is clearly ancient to the extent that it is non-specific. And you can trick the ribosome now if you mischarge tRNAs or do other sorts of things. You can get the ribosome to do all kinds of non-specific condensation. That sort of crude, ancient enzyme is still there. I mean, it's still, in some ways, even though the ribosome is the core functionality of biology, it is the sorriest enzyme that you've ever seen. There's no specific stabilization of a transition state. There's no specificity built into the catalytic site. All the specificity is added on remotely far away.
2 questions that Kamesh is asking.
Okay, so what are the other 2 questions? Can you read me? I have some more questions.
I can read you short.
Yeah.
Did you get your question answered? He says, but viral particles don't have ribosome, don't they? Ribosome-independent life. Can molecular clock and phylogenetic tree estimates point out which phyla has the most oldest ribosome? In other words, how old is the oldest ribosome? Older than 3.5 years.
Okay.
That's his first question.
So the first question was about the virus. And I mean, I think the ribosome solves that virus problem, like, is a virus alive or is it not? It's not alive until it infects a cell and gets control of a ribosome, at which point it becomes alive. So that's sort of how I would answer that question. So the other question was, which basically, the question, where are the oldest ribosomes? And the ribosome is the same age everywhere, because all the ribosomes on the face of the earth have the same ancestor, and so there's no such thing as an older ribosome or a newer ribosome. All ribosomes basically are the same age. So in fact, when we talk about the tree of life, and Carl Woese's organization of the tree of life, that actually is the tree of the ribosome. I mean, he sequenced ribosomal elements, and that's where he made that tree from. And so all that tree of life, our canonical tree of life, basically is the ancestry of the ribosome, and it all points back to the last universal common ancestor. So everything in biology has inherited the same ribosome from the same ribosomal ancestor. So they're all the same age in that sense.
Okay, I'll read his second question to you.
Mm-hmm.
RNA is always thought to be the most fragile of nucleic acids. How did the ribosome escape hydrolysis or other chemical degradation dangers?
Okay, yeah, so that question is about the fragility of RNA, and how did RNA sort of persist and be functional when it's so fragile? Yeah, that's a really good question, and in fact, that speaks directly to what the ribosome is. So the ribosome, in our view, is actually a collaboration between, I will say peptide, but possibly ancestors of peptide, and RNA and the ancestors of RNA, and the initial function of the ribosome was stability. So if you say, how could RNA survive? RNA can survive when it's protected by other molecules. In fact, the ribosome, in our view, the function of those ancient peptide precursors was to bind to the ribosome and to confer stability. So in our view of biology in general, the original, I guess you would say, symbiotic event. I mean, people think of symbiosis in terms of organism, but in our view, the original symbiotic event was the joining together of polypeptide and polynucleotide. And you say, what drove that symbiosis? And it was protection. And so the original, what we think, ancestral ribosome was actually made of RNA oligomers, not polymers. And those oligomers assembled and did a poor job of surviving until they met little peptides that could bind to them and confer stability. So that's basically that lack of stability of RNA speaks to the whole ancestral function of the ribosome in our view.
Sanjoy has a question. Is there an environmental setting that would best provide the ingredients for your view of the origin of life?
Yes. And I have to say right away that a lot of this comes from my collaboration with Nick Hudd. So we have sort of what you could say is shared views of these things, Nick and I, because we talk every day about the origin of life, and we sort of beat on each other and have reached sort of a consensus that what we think is important to the origin of life. And it might be a little different from what you read in textbooks. Number one, low water activity. There is no way life originated in a large body of water, because none of the polymers of life are stable in water. They are all synthesized by dehydration. So to us, it's obvious from looking at the universal things in biology that life did not originate in a large body of water, say a thermal vent or whatever. And you say, how could you originate life in the environment I'm talking about? To us, the critical event has got to be thermodynamic cycling, meaning basically a spinning planet or a spinning moon. But basically, that spinning moon cycles the temperature, it cycles hydration. If you really say, what is the core chemistry of biology? It is hydration, dehydration. Everything you make, every polymer you make, carbohydrate, nucleic acid, protein, all of it is made by dehydration condensation. And of course, everything that's made has to be gotten rid of, and it's all hydrolysis. And so how do you switch back and forth between hydration and hydrolysis and condensation? And a spinning planet is basically a PCR machine designed to do that on the surface. Okay, so you have every day how many droplets of water form in your front yard, or every night, and every day how many evaporate. And basically every one of those little droplets on the ancient Earth was an attempt, or you could say was an experiment to originate life, because and there was untold billions of them every 24-hour cycle. So the numbers are really, really huge.
Here's a question here.
Mm-hmm.
Oh snap! Are you suggesting an atmospheric environment or subatmospheric air and water interface environment?
I think the atmospheric environment was important in the sense that, for example, if you look at Titan, Titan, it's raining polymers on Titan, right? And I mean, so what's happening in the atmosphere is critically important. But I believe that the sort of the chemistry of assembly and the chemistry of hydration and dehydration took place on the surface, and by that I mean at the solid atmosphere interface, where basically you had hydration, dehydration, cycling back and forth. Temperatures going up and down on a day-night cycle. Hydration is going up and down, up and down. To me, if you have polymers, and you have synthesis of polymers, which actually is not hard, or let's say oligomers. I mean, obviously, the atmosphere of Titan can synthesize polymers and oligomers, so I don't think this is a really difficult thing. And it's probably obvious that the ancient environment of Earth could synthesize polymers. And then you just randomly degrade and synthesize these things by hydration, dehydration, and then sort of nothing's happened except some of them will start to assemble. And the assemblies will win. They will be less resistant to degradation. And so basically, at that point, you have kick-started evolution, and the game is on. And there's no catalysis. You don't need polymers. You only need oligomers. You don't need cells. You don't need compartmentalization. I mean, it's just on a viscous surface. So everything is simple, and it's basically unstoppable.
Sandra is asking whether, in your view, is life prevalent beyond Earth?
I would think so. In fact, I'm willing to bet that Titan, I mean, I don't know anything about life on Titan, but I bet a lot of the really interesting prebiotic chemistry will be found. And what I mean is, obviously, we have polymers on Titan, and we have liquid-solid interfaces, and we basically have drying and wetting. Now, I don't mean water, I mean hydrocarbon, but basically we have this drying and wetting going on, and we have polymers assembling, and the polymers that can assemble will win as far as the degradation fight goes. And so, I mean, to me, I think it's just something that is unstoppable.
So there was a question about your career path now. Whether you could tell us a little about your career path and how you became an astrobiologist.
Yes. How did I become an astrobiologist?
Oh, wait, hold on. Maybe before moving on that, there's another question,
okay.
Scientific question, that the most, according to Kamesh, the most striking aspect is that there are no intermediate ribosome structures in different phyla. There are only 4 subunits, 40S, 50S, 40S, 30, 50, and 40, 60 in all life on Earth. Would you have expected intermediate functional ribosomal structures in different phyla, and is life possible without water? Another question.
Okay, so first back to the ribosome and no intermediate forms. I mean, yes, of course, everything alive inherited its ribosomes from the same ancestor, so that puts kind of limits on what they're going to be. But actually, if you look at all the ribosomes, all of life, you can see that, for example, our eukaryotic ribosomes, especially metazoans, we have enormous ribosomes compared to bacteria, and so there is evolution of the ribosome, but all of that evolution is on the surface. So the way to say it is there is basically a bacterial ribosome inside of a eukaryotic ribosome. In fact, it's actually the ancestor of the bacteria and the ancestor of the eukaryote and all of the. But anyway, so when a ribosome gets bigger, which it does. I mean, if you look at what's happened in eukaryotes, you can see the ribosome has gotten bigger and bigger. But every time it gains something, it doesn't touch the thing underneath. So it really is it's amazingly similar to how a tree gains rings. The rings go on the outside without disturbing the underlying ring structure. And that rule, I mean, we have three-dimensional structures of many, many ribosomes now, and we can see that rule is very, very strict. So that basically you have the most ancient ribosomal components are preserved when you increase the size of the ribosome.
Is life possible without water?
I believe life. I don't see what's so special about water. I mean, I think life requires polymers, It requires assembly. It requires systematic synthesis and degradation. And I don't see why water is water is nice, but I don't see why other solvents can't do that. You can assemble things in non-aqueous media.
So the ribosome is really the argument against intelligent design.
Yeah, I think the ribosome is a really good argument against intelligent design, because the ribosome, obviously, if you study it for very long, you can see it is just one quick dirty fix piled on top of another, and it's just such a complicated, illogical final result that I think the ribosome argues very strongly against intelligent design. Yeah, because you never would have done it this way, for sure.
Well, it doesn't. It's my question.
There's a lot of arguments we could make.
Okay, so if any, hold on, let me ask. I don't know. I'm not sure they hear me.
Early in astrobiology career.
Astrobiology. So Batul is reading these questions to me. I
I'm
hope.
asking if there are any other questions about science in the audience.
Okay, are there any questions about science? We were going to switch a little bit over to like my career.
Okay, you can, I guess, go. Because
All right, so I'll tell you how I became a astrobiologist. I actually was not my PhD is in physical chemistry, and I studied biophysics. I did crystallography and various spectroscopies and calorimetry, and I think I got a really good training in physical chemistry, and then somehow became a biochemist, but studying nucleic acids. And I devoted my life to that, electrostatics. How does DNA bend? How does RNA fold? And I really did not think much about the deeper implications of it, I have to say. I was really thinking not about astrobiology, origin of life, anything like that. I was really just interested in DNA electrostatics. How do proteins bind to DNA? How does RNA fold? And I was very interested in electrostatic models. And then in about 2000, of course, these crystal structures of ribosomes started to come out. Tom Steitz's was the first one. And I started looking at the ribosome because it had so many magnesium ions bound to it. And I thought, if I want to understand electrostatics, I need to understand the folding of the ribosome. And I actually, it's sort of interesting, I wasn't really very interested in what the ribosome did or where it came from. To me, it was a database of molecular interactions, and I studied it in that capacity for quite some time. And then I do have this distinct recollection, I was looking at it one time, then I was studying a magnesium, and all of a sudden I thought, this magnesium has been here in this place in the ribosome for a very long time. And I started sort of thinking like that, and that was really the beginning of my road down to becoming an astrobiologist. And it was only about 5 or 6 years ago that I really sort of made the jump. In fact, I have to say, it was when our center got funded. I mean, I had these ideas, and I had these ideas about the ribosome, and but I really didn't think I was ever going to get to go after them and work on them. But then our center got funded, and so then I started calling myself an astrobiologist. And now I do experiments now that I think are totally driven by astrobiology. For example, we're interested in iron and RNA, and no one is going to be interested in iron and RNA unless they're interested in the ancient Earth and the sort of the geology. Look at the ancient geology of Earth and say, what did ancient RNA experience? So I think that really now I am in that sense a true astrobiologist.
Well, Gabriel Mikas has a question. It's not directly career related, but I'm still reading it. Whether can you tell us a little more about the endosymbiotic relation of ribosome with mitochondria?
Oh, when I was talking about a symbiotic relationship, okay, I wasn't talking about the mitochondria. I was talking about protein and RNA. So, this is the way I look at it. If you look at protein, it has a certain personality, and its personality is really all about self-assembly. And protein is I believe, the universe's, as far as I know, the universe's king or queen of molecular assembly. If you look at alpha helix, beta sheet, antiparallel beta sheet, I mean, the protein has this self-complementary backbone that is, and then if you look at the restriction of the torsion angles and everything, and that the assembly is just this beautiful sort of energetic minima of the rotation of the torsions and of the molecular interactions, and you cannot. I challenge anyone to imagine a better molecule at molecular assembly than protein. I mean, it's just unbelievable. And it assembles by its backbone. And then you turn around and you look at nucleic acid, and you can make all the same arguments. Say this is unbelievable specificity and all this, except it assembles by its side chains, and the backbone is self-repulsive. So these two molecules are as far apart as you could get. And I believe each one of them is outrageously powerful, but each one of them has horrible limitations. And so when I talk about symbiosis, I really mean these two molecules coming together. These two oligomers or polymers, but these two backbones coming together. That was the seminal symbiotic event in life, and that goes very, very deep. That's basically, to me, that's the beginning of biochemistry, or the beginning of life, is the joining of RNA and protein, which I think was very early. I don't think there was ever an RNA world. I don't think RNA ever did it alone.
Wow. So Sanjay asks, do you have an article that describes more your thoughts about life without water? This is fascinating as it goes against what most scientists think about the requirements for life, right?
Yeah, I haven't written that up, and I would not be the one to do it because I'm not doing experiments there. But you know, Nick Hudd here, they're looking at RNA folding and assembly in deep eutectics and other things. So I guess I don't have anything written up, but I think you just really need to think carefully about, to me, what is life? Life is molecular assembly. I mean, if you look at membranes, folded protein, DNA, RNA, I mean, it is molecular assembly, nominally stable molecular assemblies. That's to me the definition of life. And you say, is water required for that? I don't think so. You know, you can do it in chloroform. I mean, it's a different kind of molecule, but I don't think there's anything. You know, water has. I'm not saying it doesn't have a lot of great properties, but you can do this, you know, in other realms with other solvents. I just don't see why you couldn't. So.
Yeah, that's a wow. Any questions?
Are there other questions? Have I answered all the questions?
I think you, like, everyone's...
okay, wait, you guys, let me tell you one thing, because I guess I've said some things that are not in the mainstream. So, for example, I'm not big on hydrothermal vents, and I'm not, you know, I don't think water's necessary and all of that. And so I need to say something, which is that here at Georgia Tech, we have a fairly large community right here working on the origin of life, because we have two pretty big federally funded centers. And so we have a community of people, and we talk about the origin of life. And I think we have an advantage over some other places where we have this large community, and it's academic. We're not in a national lab, and even though national labs are great, a lot of times people at national labs are a little bit isolated. You know, you don't have this big integrated community, and so I think the fact that we have this big community I mean, we have definitely our ideas on the origin of life as a community at Georgia Tech have really sort of diverged from the consensus. And I mean, I think we're right. and I guess I think the reason why we have diverged is, number one, we're centered in biochemistry. You know, to us, if the biochemistry doesn't work, we reject it. And number two, we just have such a large community, and we really hammer on people. I mean, if I say something that Nick Hudd thinks is not reasonable, I mean, he just slams me. And so we, you know, I think we have a really good sort of environment here to think about these things in a really critical way. We've been really, really lucky here. And so you're right. I am a little bit outside the mainstream, but I think we're right. And I will refer you to Oparin. Have you guys seen this book? Where's my camera? Yes. Oparin. I would recommend this book to everyone. And I would also recommend Carl Woese. You read Oparin, you read Carl Woese, and I think you can see we are coming right down. Oparin was a plant physiologist, and Carl Woese was actually, he was almost a pediatrician. He had this kind of biological training before he went into biophysics, and I mean, to me, the people who most inspire me are Carl Woese and Oparin. Those are. And I mean, if you look at where they went, we figure out we are fitting right in between those two philosophies, and we think actually we can bridge Oparin and Carl Woese. That's basically where we're looking.
Question is, what is your suggested academic path to study the origin of life?
Well, number one, I think to study the origin of life, I think, you need a discipline. You need to, you shouldn't say, I'm going to study the origin of life. You need to have a well-grounded discipline of some kind, a traditional discipline, I guess is what I mean. But to me, it's biochemistry. I mean, I think, geology is important. It informs it. But I think biochemistry, because the origin of life is basically, to me, it's the oldest biochemistry. That's basically what we're asking, and it's not geology. I mean, geology is important. You have to know, what was there, but we're talking about solution chemistry. We're talking about the most ancient biochemistry. So I would recommend biochemistry, personally. Of course, I'm a biochemist. So I'll admit a little bias. But no, originally I was a chemist. I was a physical chemist. But if you read Oparin and you say, how did he get that? How did he come up with all of that? He came up with it coming up through biochemistry. That's where it came from.
Where do you see the commonness eye to biochemistry?
Sorry?
Some people second your thoughts about biochemistry being essential. How do you see geology or other studies in the study of original?
Can I be frank? I'm going to be frank.
Keep in mind that they're geologists.
I know. Look, in this world, we have big disagreements, right? But we might as well be frank, which is that I think geology, obviously it's very important. In fact, geologists, I gave a talk once on RNA and magnesium, and a geologist came up to me and said, you need to look at iron. And I scoffed at him. And man, was he right. I mean, so I'm not saying that, all of these disciplines can't go. But the idea that the origin of life was a geological event, I think, and that, you have to explain it in terms of, if you look at the explanations in the thermal vents and basically that the origin of life is being explained in terms of geology, I think that's a wrong path. And I think that's, you look at Titan. Okay, it's raining polymers on Titan. This is not geology, okay? This is chemistry. And, I think the origin of life on Earth, I think, that's the kind of thing you have to understand, is that chemistry of where do those molecules come from. It's raining amino acids down on our heads right now, right? This is not geology, right? We don't have to say that thermal vents made amino acids. I mean, they're just raining down on our heads as we speak.
So Aditya Chopra notes that it's maybe geochemistry. Like, how about geochemistry?
Well, yeah. I mean, it's hard to know what things are, but okay. I mean, this is my bias. I'm a biochemist, and when I look,
we're all one big happy family.
We're all one big happy family. But when I look at, I say, what is life? Life is not, inorganic chemistry. Life is assembly. It is membranes, and it is proteins folding. It is that. And you say, where did that assembly come from? Did that come from geology, or did it come from organic chemistry? I mean, of course, there's geological influences, but I think you really got to look at the organic molecules that were available, and the Earth was slimed out of them. I mean, there are dunes of polymers on Titan. Dunes. I mean, imagine the Earth before life. There was so many things to eat. Okay, it was you don't have to worry about where these molecules came from. The Earth was slimed with them. It was 5 feet deep, just like Titan. And so then you just have to say, then what happened? Things started assembling. And that assembly is still imprinted in biology. And things started, there was hydration, dehydration, and that hydration, dehydration is still imprinted in biology. All of that origin of life chemistry is still here.
Well, I should read the note that Sanjoy said. Well, so people are throwing words here, that geobiochemistry, they're not words, but the meaningful words. Geobiochemistry, astrogeochemistry, maybe the origin of life is an interdisciplinary endeavor. And also a question that this is a valid observation, a comment. Many geologists have blinders on when it comes to diversity of environments, microenvironments, and alternative geochemical regimens that could have existed on the Archean and Hadean Earth.
Yes. Right. Yeah. Okay. So yes, there's a question of microenvironments, and yeah. And let me just say this about for a person like me, what I would like to do is go to a geologist and say, okay, what was the Earth like, 4.1 billion years ago, or whenever we think life, and the problem that we all have is that there are models for the ancient Earth, and they're based on sparse data. And that's the thing we have to remember, is that even the atmosphere of the Earth, we really don't know. If you have a model for the origin of life that requires CO2, or you have a model of the origin of life that requires some hydrogen, or some specific thing, then I think you have a tough road to hoe, because it's going to be very difficult to really nail down what those conditions are. And I think what we want is robust models that are sort of independent of that. I mean, we know the Earth was turning. We know the sun was shining. There are certain things we know, and if I don't want to try to come up with models for the origin of life that are dependent on CO2 or that are dependent on hydrogen or something, because at this point, I don't feel with the atmospheric, or that the people can really tell us, or even, was there land or was there no land? I mean,
so Sanjoy asks, actually, if life occurred on the surface, what atmosphere do you think was about to fuel it?
I think it's probably pretty clear that there was no oxygen, or there was very little oxygen. It was a very reductive atmosphere. I think most people will concede that. But other than that, actually, I don't care. I don't find it that interesting a question. I don't find it to be, at least the way I look at the origin of life. I mean, there were amino acids, right? Amino acids are made in outer space. They're raining down on the Earth. It doesn't matter what the atmosphere of the Earth was. There were amino acids here. And I mean, to me, that's the kind of logic we need to follow. I don't want to have a favorite environment or a favorite microenvironment that my whole model is dependent on. That's another reason why I'm not so big on the thermal vent model, is that the statistics are against you, in the sense that it requires a very specific sort of environment. You have to have, I don't know, sulfide or whatever. And I mean, I just think the origin of life was very robust, and I just think it happens under a variety of conditions. I don't think it's that condition dependent. I just think if you have a spinning planet and you have organic molecules, you get life. I mean, it's
Sandra is noting whether nitrogen seems ubiquitous.
Yeah, nitrogen. I mean, obviously you need carbon, nitrogen. You need the elements to make the polymers. You need that rich organic chemistry. To me, it's an organic chemistry problem, but it's not. Sometimes, and I'll say something else. Maybe this will make people mad too. Is people talk about energy and they say, well, where did the energy come from? And to me, this is also an uninteresting question, because if you have a spinning planet, it doesn't matter if it's the Earth or Titan, you are never at equilibrium, right? You are always pushed away from equilibrium. You have the temperatures going up and down, and with that, you don't have to worry about energy. I mean, basically, you're always moving. Basically, there's a driving force for things to happen. And when people talk about energy, I think what they're really saying is thermodynamically linked chemical reactions. That's what I think they really mean. I mean, this is my own interpretation of what they mean. And whereas I think if you look at people like me, I would say the chemical reactions don't have to be thermally linked. I don't care if the amino acids are made in asteroids, rain down on the Earth. I don't care about linking their synthesis as long as they're made. There's no thermodynamic linkage between their synthesis and what happens to them. So I find the energy question to be uninteresting.
So Aditya Chopra, actually from Australia, he says the present day or the early Earth is not or cannot be a homogeneous environment. It is a heterogeneous environment, so a mix of reactions could take place. I see Loren's point, but note that it is not possible to decouple environment from the reactions. And he notes this is often a big critique of many of today's lab efforts to recreate the origin of life.
Well, I'm not saying the environment is decoupled. I think the environment was absolutely coupled to the origin of life. In fact, I think without a spinning planet, no life. Okay, so, but, because you definitely need an energy flux. You need to be pushing, if you're at equilibrium, you're dead, and you're never going to originate life at equilibrium. So you say, how do I get out of equilibrium? Is a spinning, that's what a PCR machine is. You're just cycling things back and forth, and that cycling keeps things out of equilibrium. And so, of course, and the other thing is if you look at the kinds of molecules that will be incorporated into biological systems, of course that is going to be linked to what's there. So I'm not saying there's no linkage, but I think, my sort of, at least the way I look at it, I just think that life could originate under so many different environments. I don't think it is dependent on, I think it's probably dependent on amino acids, actually, because it's hard for me to imagine something other than protein. So obviously there are a few sort of basic things like that, but I don't think there's too many. I don't think there's too many of those things.
Which I think Sandra's question is a good time to ask. What are your thoughts on life originating on Mars?
On life originating on Mars? Well, if Mars was warm and wet, I don't know why it wouldn't. Yeah, I'm high. And the question is whether there's still life on Mars, buried down in the ice somewhere. That's kind of an open question. But I think life originates quickly, and I think it's just chemistry. It's nothing. You don't have to look at. It's not something exotic. It's something almost inescapable if you have the right conditions. And I don't see why the conditions were not right on Mars.
Okay, Gavril Mikas is asking what kind of biochemistry could favor ribosome development in a Martian cave, for example.
In a Martian cave?
Mm-hmm. Biochemistry that could favor ribosome development.
Well, okay. First, I'm going to redefine the ribosome for you. I'm going to define the ribosome as cooperating polymers. That's my definition of the ribosome. The ribosome is the place in biology where protein and nucleic acid comes together. And to me, that is sort of one of the necessary for the life we see around us. You've got to have multiple kinds of polymers, well, two, that do it. And so you say, could that happen in a cave on Mars? I would think not in the cave totally, but things have to be happening outside the cave too. There has to be a supply of monomers. There has to be, I would think, cycling, hydration, dehydration, and things like that. So I would say not in a cave, but outside a cave. But I don't see why not. I don't think necessarily on current Mars, but on ancient Mars, yes. I don't have a problem with it.
Yeah, I should note that Gavril is from Greece. That's the international crowd we have today. And let me see if there are any more questions. Loren, you're very these are very big statements. I think everyone is.
Usually I don't say these things because people get upset, but I thought, what the hell? You guys don't repeat anything I'm saying, okay?
It's too late. We've been live tweeting what you're saying.
Oh, shoot. And no profanity.
Could you tell the title and the authors of the book you recommend? I can write them down. Actually, if you just hand them to me.
Well, there is this Origin of Life by Loren. Oh, my camera's over here. Sorry. This, I think, is really an amazing book. And obviously, you know, you have to realize that this book is 100 years old, and there's things in it that are obviously wrong. But I think this is a biologist or a biochemist, a plant physiologist, trying to explain the origin of life. I think it's many, many things right. And then I would recommend some of Carl Woese's papers. There's one, I think, from 2002. And I can really briefly tell you, you know, if you look at horizontal gene transfer, for example, and this is something that Carl Woese really looked at carefully, you can see as you go backwards in time, the rates speed up. And you say, why would this be? Why would horizontal gene transfer depend on time in this way? And the idea that Carl had, I don't know if it was his original idea, but I think maybe it was that as systems become more and more complex, it becomes more and more difficult to interchange parts. And so you go back and you just project this backwards, and you could say the last universal common ancestor, that was the point at which horizontal gene transfer, or just transfer, got slow enough to allow speciation. So before that, you had diversity of life, but there was this smearing process because things were simple and components. It's like you have a Model T and a Model A. You can change the generator between them. I mean, that was basically what was happening. You had very primitive biological systems, and so there was no speciation because as soon as something diverged, it would get kind of folded back in and averaged out. And then you reach a level of complexity where the transfer got slow enough, and all of a sudden you got speciation. And so, but keep walking that logic backwards and say, okay, I'm going to keep moving backwards, and I'm going to keep simplifying, and I'm going to say, what were the elemental parts of life? And that's where, you know, I think if you really say that life could have originated possibly with few components, and it was where they started exchanging and where different polymers met each other and could work together. So that we're taking Carl Woese's logic all the way back to the origin of life and say, what does this really mean for the origin of life? It was where these two polymers could come together and they could exchange functions. And that was the origin of the ribosome, and that was the true origin of life.
The question is by Aditya Chopra. Any guess and picks for tomorrow's chemistry Nobel Prize?
Yes, Ned Seaman. Or maybe M.G. Finn.
Hey, he's from Georgia Tech.
M.G. Finn is here at Georgia Tech. And Ned Seaman is an old friend of mine who is... so I'm biased. But no, Ned Seaman invented the whole world of DNA nanotechnology. And he did it alone, and he did it against great odds and with very few resources. And he is just an unbelievable scientist. And I mean, and he's got some recognition, but not what he really deserves. He's created an entire field of science, in my view.
Well, he got nominated for a Nobel Prize. That's a recognition.
Yeah, well, he needs to have a Nobel Prize.
Or you get to have a friend who has a Nobel Prize.
Yeah, I want my friends to get Nobel Prizes.
Yeah, so DNA nanotechnology is also getting involved in the chat room.
Yeah.
Any last words of wisdom, Loren?
You know, I don't know. For young scientists, do I have words of wisdom? I have to say, be tough. You know, we're not in good times. And, but I think things cycle. I mean, obviously everybody, I guess, I don't know about all of you, but this whole shutting down the federal government and the whole way things have happened, I just find it so discouraging. But being a scientist is so fun. And I don't know if you guys can tell, but I love being a scientist, and I love thinking about molecules and the origin of life, and that it is worth some risk, but I just say that things will cycle. I mean, I believe my father got a PhD in 1970, and I mean, it was very bleak. That was Richard Nixon was president, NASA was hacked, everything was terrible, and then it came back, you know, so we're kind of like that maybe, and things will come back.
Aditya is asking on the movie you were in, on the origin of life, the one that was broadcasted at Upcycle, whether that is released or is it on YouTube?
I don't know. That's actually in possession of Montana State and the NASA Astrobiologist. I don't know what has happened to that meeting. Actually, I was wondering what happened to that. I don't know. I'll find out and send you email.
I
But
heard
did
that
you-
it was broadcasted on the planes in international flights.
Really?
Yeah.
Okay, so it went somewhere.
So go buy a ticket.
Yes, so go ahead and buy a ticket.
As they send you out of the country, they show you that movie and don't let you come back.
Yeah, so do you want to make? I mean, if there are no other questions, we'll just ask Loren to maybe say a few last words again on anything that you want to share with the young people here, and this is being recorded.
Okay, this is being recorded. He's telling me to be cautious.
So it's too late for that.
I know. Gosh, yeah. Let me just say a few things about the origin of life. I am new to essentially, I've only been really thinking seriously about it for maybe 5 or 6 years. And so in some ways I come in with sort of not baggage that, this whole argument people had about, metabolism and information and all that. I didn't even know about any of that. And sort of went through most of my life without understanding any of this stuff. But what I do see in the origin of life is that, and I'm at risk of this too, is that people become enamored by, in fact, you could accuse me of it, and I would say touche, of, I'm a geologist, I want geology to be important, I'm going to study vents, or I'm a biologist, I want the ribosome to be important, or whatever. I mean, that is something that I think plagues our discipline, and that if we really want to make progress, basically, we have to do experiments. And so, you cannot believe how many papers I get to review models for the origin of life about the ribosome, and I write the same review every time. I just say, propose an experiment that will test this model, and if there's not an experiment, then the model has no validity, and just throw it away. I don't say it quite so meanly, but I mean, that's really what we have to all hold ourselves to, that standard. And we have to also say, is there an experiment I could do which would cause me to cast my model away? And if there's not, then there's something wrong, right? I mean, so there has to be a definitive yes or no experiment to these various models, and we need to do those experiments, and we need to use them to move forward. So for us, the experiments are making the ancestral ribosome. Can we actually recapitulate these events? Can we do the assembly? Can we do, I mean, in my model, I can start replicating models based on assembly, replicating molecules based on assembly. Not replication in the sense of a DNA polymerase, but achieving the effects of replication. And so I have to do those do those experiments. And if I can't do them, then I should shut up. That's basically, or not, I don't have to do them, I have to talk somebody else into doing them. But somehow the experiments have to happen. And all of us need to hold ourselves to that standard in this area. And I think that's the main thing we need, is to basically, when we talk about the origin of life, we need to say this is an experimental science, and we need to push it forward by experiment that we can use to refine our models and also to get rid of models. So that's my philosophy on it.
Well, I guess yeah, we will end here because it's been an hour. And,
okay. Well, I've enjoyed talking to you guys. You don't have to clap if you're sitting by yourself in the computer. No one can hear you.
But yeah, thanks everyone for being here.
All right. Thank you guys for listening to him.
Thank you.
Thank you. I enjoyed this. I enjoyed it a lot. Okay. Don't tell on me.
All right.
All right.
So
bye.
thanks everyone. Bye bye.